Hello students, I hope you all are good. Well, in this video lecture, we are going to start the very first kingdom, that is kingdom Monera, which is part of your second chapter, that is biological classification. Okay. Well, so let's start learning about kingdom Monera. Now, if we talk about kingdom Monera, this kingdom is consisted of all prokaryotic organisms. Okay. And all of these prokaryotic organisms are unicellular. Okay. Now, what I mean to say is kingdom Monera is comprised of all prokaryotic organisms. Okay. Now, this is a fact that all prokaryotic organisms are unicellular. Therefore, kingdom Monera is comprised of unicellular prokaryotic organisms, and all these prokaryotic organisms are collectively known as bacteria. Okay. Now, since all prokaryotic organisms are known as bacteria, it means kingdom Monera is only comprised of bacteria. Or in other words, we can say that the sole member of this kingdom is bacteria. Okay, याद रखिएगा ये कई बार पूछा गया है कि bacteria is the sole member of kingdom Monera. Okay, so keep a note of it. Great. So kingdom Monera comprised of all prokaryotic organisms which are collectively called bacteria. Now, since these all are prokaryotic organisms, let's see what are their features. Okay. Now, I'm sure all of you must be knowing the difference between prokaryotic organisms and eukaryotic organisms, or you all are familiar with prokaryotic and eukaryotic cells. Okay. Now, whatever difference you have studied about prokaryotic and eukaryotic cell, the same holds true for the difference between prokaryotic organism and eukaryotic organism. However, we are going to revisit them. So the first feature that we are going to talk about the prokaryotic organism is about their nucleus. Okay, before we do that, let me reiterate. See, in biology, you should know how to split the terms to to understand the meaning of the term, to derive the definition of that particular term. Okay, so if you start doing this, you will not forget. Okay, if you keep mugging the definition of these terms, you might forget. Okay, but if you know how to split them and understand the meaning of the whole term, then it is very difficult that you will forget them. Great. So taking example of prokaryote here. Now, prokaryote or prokaryotic, this word is made up of two words, pro and carrion. Okay, ध्यान दीजिएगा, pro and carrion. Now, pro means primitive and carrion means nucleus. So all those cells or the organisms which have primitive nucleus are known as prokaryotic organisms or prokaryotic cell. Okay, great. Now, why I'm using this cell and organism interchangeably? That is because the prokaryotic organisms are unicellular. So you can treat them either as one cell or as one organism. Both are the same thing. Okay, great. So when we are saying that these organisms or these cells have primitive nucleus, what is primitive nucleus? In what sense this nucleus is primitive? Let us see. Now, such nucleus do not have, or such organisms do not have distinct nucleus. Okay. Now, why not? Because this nucleus, okay, does not have nuclear membrane. They lack nuclear membrane. They lack nucleolus. They lack histone proteins. Okay. So all those, just a minute. So all those cells which have primitive nucleus, or those who do not have distinct nucleus, they lack nuclear membrane, they lack nucleolus, and they lack histone protein in them. Okay, and such a nucleus which is lacking all these three is known as primitive nucleus or incipient nucleus, or we also call them nucleoid or genophore. Now, nucleoid, wherever this word OID is used as a suffix, this means like. So nucleoid, this word means nucleus like. It is not the complete nucleus, but it is just nucleus like. Okay, or genophore. So whenever you see a prokaryotic cell, you will find that inside that cell there is no distinct nucleus. Rather, the DNA is just lying around into the viscous fluid which is present in the cytoplasm, and this area of cytoplasm having viscous fluid with the DNA inside it is considered as a nucleus. Okay, it is considered nucleus like structure and hence called nucleoid or primitive nucleus or incipient nucleus or genophore. Okay, and the organisms which have this primitive nucleus are known as prokaryotic organisms. Okay, and these primitive nucleus lack nuclear membrane and hence they are not very distinct. They also lack nucleolus, they also lack histone protein, and that's why we do not find the typical chromosomal formation, the X-like structure formation in case of prokaryotic organisms. Okay, we will see these things in detail when we will be talking about we will be talking about the cells and different types of cells in the corresponding chapter. Okay. Now, apart from the nucleus, the prokaryotic cells also lack membrane bound cell organelles. Okay, so all the prokaryotic cells do not have membrane bound cell organelles, any of membrane bound cell organelles, whether it is single membrane bound or double membrane bound. No. However, this does not mean that they do not have cell organelles at all. They have. If there is a cell membrane which is naked or which is not membrane bound, will be found in prokaryotic cells as well as in eukaryotic cells, for example ribosomes. But all the organisms which all the cell organelles which are membrane bound will only be found in eukaryotic cell and will not be found in prokaryotic cells. Okay, let's see. So they do not have membrane-bound cell organelles like mitochondria, chloroplast, lysosome, endoplasmic reticulum, Golgi body, you know, rest of any of the microsomes, etc. However, see, the mitochondria is very important for survival because it is the powerhouse of the cell where ATP is generated, and even the prokaryotic cells require ATP. And if they do not have mitochondria, then where this ATP is formed? And if it is not formed, then how the prokaryotic cell is surviving, right? So in prokaryotic cells, they do not have membrane-bound organelles like mitochondria, chloroplast, or lysosome, but they have their analogous structure. Okay, analogous structures are the structure which are structurally dissimilar but functionally similar. Okay, so see, there is a structure in prokaryotic cell called mesosomes, and these structures are analogous to mitochondria, as in mesosomes ATP synthesis occurs in case of prokaryotic cells. Okay, we will see later in this particular video lecture how the mesosome look. Okay, similarly, in place of chloroplast, the prokaryotic cells have a structure called chromatophore. Okay, so chromatophores are analogous to chloroplast, and it is only found in the autotrophic or photosynthetic bacteria or Moneran member. Okay, Moneran as in the member of kingdom Monera. Okay, so remember, in prokaryotic cell, membrane bound cell organelles are absent. However, there are some special structures which are analogous to some of the membrane bound cell organelles. For example, mesosome is analogous to mitochondria, chromatophore is analogous to chloroplast. Let's see the other one. The other one is called periplasmic space. Now it is not an organelle, okay? It is not an organelle. It is a space, okay? It is periplasmic space, and this particular space contains various hydrolytic enzymes. As a result, it is analogous to lysosome, okay? Just remember these things. You may not be able to appreciate it completely in this video lecture, but slowly as we move ahead, I'll assure that you'll have a better knowledge and you'll start appreciating these things slowly. Okay, so let's see how these things look, how a typical prokaryotic cell looks like. So if we draw it, so what I'm doing is making a typical prokaryotic cell for you. Let's see here. So this is a typical structure of a bacterial cell or a bacterial organism. What you find, see, the white line that I draw is cell membrane. Okay, let me do a recap of these things. Okay, so this white line, what I have made here is cell membrane. Okay, and this cell membrane is actually plasma membrane. It is similar to the plasma membrane what we found in eukaryotic. I'm saying similar, it is not identical to the eukaryotic plasma membrane, but it is similar to the eukaryotic plasma membrane. As in, this cell membrane or plasma membrane in prokaryotes is also made up of lipid bilayer protein and cholesterol-like substance. Okay, great. Now, apart from this cell membrane, you find that outside this cell membrane, there is another layer which is called cell wall. Okay, so this yellow line represents a cell wall, and in case of bacteria or prokaryotic organisms, the cell wall is not cellulosic. We have studied earlier. Rather, it is made up of peptidoglycan. Okay, peptidoglycan because it is a composite or it is a hybrid molecule which is made up of protein and carbohydrate. So peptido for protein, glycan for carbohydrate. So when one carbohydrate and protein are covalently linked to form a single molecule whose one part is carbohydrate, the other part is protein, then such a molecule is called peptidoglycan. And in case of bacteria, the cell wall is primarily made up of peptidoglycans. Okay, outside the peptidoglycan, I have drawn this pink layer. This represents the capsule. Okay, great. So capsule, cell wall, and cell membrane. As I said, the cell wall here is made up of peptidoglycan. Correct. Now, once we move inside the cell membrane, what we find that inside the cell, there is this thread-like DNA structure which is coiled. Now, this large DNA is called chromosomal DNA. Okay. One more thing about chromosomal DNA. Remember, in case of prokaryotes, or we say prokaryotic chromosomal DNA is double-stranded and circular. Okay. Now you must be saying or thinking that you have drawn something weird, some entangled structure, and you're saying that it is circular. It does not appear circular. Okay. What I mean here is it is a complete circular structure, as in there is no end to it. Okay, it is a big circular thread-like structure which has been coiled to take such a shape. Are you getting it? Great. So the chromosomal DNA or the prokaryotic chromosomal DNA is composed of, or it is made up of, double-stranded DNA thread which is circular, as in it has no free ends. Okay, great. So this is circular DNA. Very often you will find small d and small s written, which represents double-stranded, and then circular. So DS circular DNA is prokaryotic chromosomal DNA. Great. Now, apart from that, there are some other circular DNA, small circular DNA. Okay, as I'm drawing here. So there are other circular DNA in the cytoplasm which are known as extra chromosomal DNA, okay, because they are extra pair, okay. So extra chromosomal DNA, and these extra chromosomal DNA are commonly known as plasmids, okay. Now, what is the main difference between chromosomal DNA and extra chromosomal DNA is that chromosomal DNA is mandatory, it is essential. If there is no chromosomal DNA, the bacteria would not survive, it will die. But if there is no extra chromosomal DNA, the bacteria can still survive, okay. So for survival, chromosomal DNA is crucial. Extra chromosomal DNA is auxiliary, okay. It aids, as an extra chromosomal DNA or the plasmids have information which helps or give advantage to the bacteria in some or the other natural conditions, okay. But it is not mandatory for survival, it is not crucial for the survival, okay. So just remember, we will talk about plasmids and different types of plasmids in detail, and then you will better appreciate this whole thing. Great. Apart from that, what we find is this entire area is filled with a jelly-like fluid, that is cytoplasm, and this cytoplasm is not uniform everywhere, okay. What we find is around this chromosomal DNA, this fluid is much more dense, okay, or you can say it is more viscous, right? And this viscous fluid, this viscous jelly-like fluid around this chromosomal DNA makes the whole structure, that is chromosomal DNA plus this viscous fluid around this DNA, appear as a nucleus-like structure which is known as nucleoid or primitive nucleus or incipient nucleus or genophore, okay. Now see, this particular thing is not isolated from rest of the cytoplasm by a membrane or by nuclear membrane. समझ में आ रहा है? And that's why we call it nucleus like structure or primitive nucleus, okay. We do not call it proper nucleus. Are you getting it? Great. Okay, now apart from this, inside the cytoplasm we also find some cell organelles, okay. So cell organelles like I'm drawing here is my ribosomes, okay, because ribosomes are membrane-less cell organelles, and as I have said that prokaryotic cells have membrane-less cell organelles, just they do not have membrane-bound cell organelles, okay. Now in bacteria or in prokaryotic organisms, the ribosome found is different from that in eukaryotic organisms, okay. In eukaryotic organisms, what we find is ATS type of ribosomes, but in prokaryotes, what we find is 7TS type of ribosomes. What are they? We will study in the chapter cell, okay. So don't worry as of now. Now, one more thing that I want to bring about is, can you see the space between cell membrane and the cell wall? Now, this space between cell membrane and cell wall is known as periplasmic space, okay. And this periplasmic space is filled with a fluid which contains hydrolytic enzymes, and since these are hydrolytic enzymes, they can degrade anything, okay. And that's why this periplasmic space is considered to be analogous to lysosome, okay. Great. Now in bacteria, at many places what we find is the cell membrane folds inside, okay, inside as in on the cytoplasmic side, and it folds to form these lamellae like structures which are known as mesosomes, and these mesosomes contain the enzymes for aerobic respiration. As a result, the aerobic respiration in bacteria takes place in the mesosomes, and as a part of aerobic respiration, ATP is formed here. ATP is synthesized here. And since ATP is getting synthesized in the mesosomes, we call it analogous to mitochondria. Okay, great. Apart from that, we also see that at some places, the cell membrane again shows infolding or folding towards the cytoplasmic side or the inner side to form chromatophores. Okay, now these chromatophores, as I said earlier, are analogous to chloroplast. Why? Because they contain the pigments, the photosynthetic pigments, and they help in carrying out photosynthesis. Okay, however, let me tell you that this photosynthesis is different from what we see in higher plants. Okay, also the pigment is different. In higher plants, the photosynthesis is done by the help of chlorophyll, but in bacteria it is done with the help of bacteriochlorophyll. Okay, and this bacteriochlorophyll is significantly different from the chlorophyll. Okay, so I hope you have a basic idea of prokaryotic cell, how they appear, what are different structures found in them. Okay, slowly as we move, we will learn more about this bacteria and different structures of a prokaryotic cell. Okay, great. Now let's talk about some other characters of kingdom Monera. Okay, so what we are going to see here is habitat. Okay, so what is the natural place where the members of kingdom Monera are found? Okay, I hope you all know habitat. Habitat is the natural place where an organism lives and carry out its physiological and ecological role or function. Okay, so that place is called habitat. Say for example, uh, the habitat of a deer is a grassland, right? A habitat of fish is water, correct? Habitat of whale is salty water, right? So habitat. Now, similarly, if we talk about habitat of kingdom Monera, what we see is kingdom Monera is comprised of bacteria, correct? So if we know where these bacteria are found, that will be the habitat for kingdom Monera, and we know that bacteria are found everywhere, in water, in soil, in air, okay, everywhere. So habitat for kingdom Monera is cosmopolitan. Why? They are found everywhere. That's why cosmopolitan, or you can say they are ubiquitous. Ubiquitous means they are found everywhere. Okay, so when somebody asks you the habitat of kingdom Monera, it is cosmopolitan or ubiquitous. Okay, they can be found in any place. Great. Apart from habitat, if we look at the mode of nutrition, the kingdom Monera, the members of kingdom Monera, that is bacteria, can be autotrophic or they can show autotrophic nutrition. In autotrophic nutrition, they can show photoautotrophic nutrition or they can also show chemoautotrophic nutrition. Okay, great. In a while, we will talk about these terms. Apart from autotrophic nutrition, the bacteria also show heterotrophic nutrition, and within heterotrophic nutrition they can show holozoic, they can show saprotrophic, they can show parasitic. So what we see here is the members of Monera are showing all different kind of mode of nutrition possible, right? In fact, kingdom Monera shows the maximum nutritional diversity. Okay, so kingdom Monera shows maximum nutritional diversity, and this question has been asked multiple times, so keep a note of it. Okay, now before moving ahead, let me talk something about the photoautotropes, chemoautotropes, and things like that. Okay, see, until now, up in class 10th, what you have studied is autotrophic and heterotrophic mode of nutrition. Okay, now autotrophic is the kind of nutrition in which the organism prepare its own food by the process of photosynthesis, okay, from inorganic materials, inorganic raw materials, I would say. Let me modify this definition for you. Autotrophic nutrition. Okay, again, before doing that, I'm splitting this term. Auto means self and trophic means food. Okay, so autotrophic mode of nutrition is a type or mode of nutrition in which organism is going to prepare the food for himself or herself, right? Self and food. So the organism is going to make the food for themselves. From what? From inorganic raw materials. So any organism which is capable of making their own food from inorganic raw materials are known as autotrophic, or such mode of nutrition is known as autotrophic nutrition, and the organisms are known as autotrophs. Okay, great. So you'll say it is the same thing. How it makes difference? It makes difference as in what you have studied so far is autotrophic nutrition always use light as energy. This is what you have the notion. It is not so. Okay. For making food from inorganic raw materials, energy is required, and if this energy is taken from light, then such a process or such a mode of nutrition is called photoautotrophic nutrition. Okay. So is there possibility of something else? Yes, there is. There are organisms which can show chemoautotrophic mode of nutrition, as in to convert the inorganic raw materials into energy rich complex organic compounds or food. They take energy from chemical reactions. Okay, you know there are chemical reactions which are exothermic. So if there is a chemical reaction, exothermic chemical reaction, energy will be released, and using that energy, such kind of bacteria can convert the CO2 into glucose, that is inorganic carbon into organic carbon, inorganic raw materials to organic food. So this is also what? Autotrophic mode of nutrition, but here the energy is coming from the chemical reactions, and that's why it is called chemoautotrophic nutrition. Okay, समझ में आ रहा है? So photoautotrophic, chemoautotrophic. In some other lecture we will split it further. Okay, now photoautotrophic nutrition or photoautotrophic reactions can be oxygenic or anoxygenic. So oxygen can be liberated, oxygen cannot be liberated. That is also a possibility. Okay, and again the chemoautotroph can be further divided. So that is a bit more detail of different kind of autotrophic nutritions or autotrophic processes that are found on our planet. Okay, but that we will discuss in some other lecture. However, what I want to emphasize here is the members of bacteria can show all different possible types of autotrophic nutrition, as well as all different possible type of heterotrophic nutrition. Okay, and that's why what we find is this kingdom Monera shows the maximum nutritional diversity. No any other kingdom has as wide nutritional diversity as Monera have or Monerans have. Okay, great. Well, so once we have seen this, the basic structure of the prokaryotic cell or prokaryotic organism, let's see what are different types of prokaryotic organisms. Okay, so if we are talking about prokaryotic organisms, they can be divided into the following groups. Okay, and that is archaebacteria, eubacteria or true bacteria. There can be cyanobacteria, which are also known as blue-green algae, or can be actinomycetes, which are also known as ray fungi, or can be a member of rickettsia, or maybe mycoplasma. Mycoplasma. Great. So the prokaryotic organisms can broadly be divided into these 6 categories: archaebacteria, eubacteria, cyanobacteria, actinomycetes, rickettsia, and mycoplasma. See, what I'm trying to say here is, when we are going to study this kingdom Monera, the study will be completed only if we study about these 6 different type of organisms. Okay, Great. So this we will see slowly when we proceed into this chapter. Okay. So to start with, we are going to first study the first group, that is eubacteria. Okay. Now eubacteria or true bacteria, this is the group that we are going to study in, in comparatively detail as other type of prokaryotic organism that I just made you count. Okay, so the most important one is eubacteria and archaebacteria. These two we are going to see in more detail. The rickettsia and actinomycetes and mycoplasma, we only have to get a general idea about them. Okay, so this is what we are going to do. We are not going to overburden ourselves with information. We are only going to study what is required, what is relevant for NEET as well as your board exams. Okay, this is a good strategy. Great. So eubacteria, correct, or true bacteria. The eubacteria can further be classified into different groups on the basis of their shape. Okay, so if we are talking about shape, the eubacteria can either be coccus. Okay, so the shape for the coccus bacteria is spherical. So any eubacteria which is spherical in shape is known as coccus. Now coccus is singular. The plural is cocci. C-O-C-C-I. So cocci is plural, coccus is singular. Do remember these things. Okay. So if the bacteria is spherical in shape, it is called coccus or cocci. If it is rod shaped, then we call it bacillus or bacilli. So rod shape is bacillus. If the bacteria is comma shaped, then we call it vibrio. And if it is spiral in shape, we call it spirilium or spirilla. Okay, so spirilla or spirilium if it is spiral in shape or spring-like. Okay, if it is comma shaped, then vibrio. If it is rod shaped, then bacillus. And if it is spherical, we call it coccus. Okay, now suppose I ask you, what is the shape of E. coli? Or I would ask, what is the shape of lactobacillus, right? The bacteria that is found in curd. Now lactobacillus, since it is a bacillus, the shape would be rod shaped, isn't it? So once you know the technicality, it's easy to remember. So lactobacillus is rod shaped. Similarly, if I say the pathogen of cholera, what is its shape? Now, if you know the name of the pathogen of cholera, that is Vibrio cholerae, you get the answer, right? It is Vibrio. So it will be comma shaped. Correct? So just keep thinking and you'll get a lot of ideas. Okay, so the eubacteria on the basis of shape is mainly divided into these four categories, that is coccus, bacillus, vibrio and spirilium or spirilla. Now further, the coccus and bacillus groups are divided. However, vibrio and spirilium are not that much divided, or at least we do not have to study in class 11 and 12. That is out of the scope. So let's see. The coccus bacteria are further divided as diplococcus. Okay, so suppose there is a spherical bacteria which is one cell, one organism individually there. It is monococcus, that is plain coccus, simple coccus, that is okay. What if a particular species of coccus bacteria always lives as two cells together, right? As I have seen here, as I have drawn here, can you see there? So if there are two cocci always linked to each other, then we call it diplococcus. Okay. What if four of them? We call them tetracoccus. Easy. Now, the tricky part. If there are 8 of coccus in a group such that they are arranged in a cubical fashion. Okay, you see the cube. So if there are 8 coccus bacteria arranged in a cubical fashion, then we call it sarcina. Okay, just remember it. So sarcina, it is unique one. Apart from that, if the coccus are arranged linearly to form a chain, we call it streptococcus. Okay, if they are arranged in a group, okay, or in a grape-like bunch, then we call it Staphylococcus. Okay, so if the coccus or cocci bacteria are arranged in a grape bunch-like fashion, we call them Staphylococcus. If they are arranged linearly to form a chain, Streptococcus. If they are arranged in a cube-like structure, Sarcina. If they are arranged in a square-like structure, Tetracoccus. If two are arranged, then Diplococcus. Easy? Great. Similarly, the bacillus is also divided on the basis of how they are arranged. Okay, if it is single bacillus, then it is okay. If two of them are arranged, then it is called diplobac or diplobacillus. Okay, now if there is a chain of bacillus, okay, linear chain, then we call it streptobac or streptobacillus. Can you see some similarity? If chain-like arrangement is called strepto. If it is chain-like arrangement of a spherical bacteria, then is streptococcus. If it is chain-like arrangement of rod-shaped bacteria, then it is streptobacillus or streptobac. Easy, right? Okay, great. If the bacillus are arranged one above another like a stack, then we call it palisade. Then we call it palisade. Okay, so these are the main categories of eubacteria or bacteria on the basis of their shape. Okay, now let's see the classification of eubacteria on the basis, or bacteria on the basis of flagellum. So if we were to divide the bacteria on the basis of flagellum, the first type we find is Atrichus. Now A means no, and trichus word is used for hair-like structure or thread-like structure. Okay, hair is trichus. It is trichology, study of hair. Okay, so flagella is like hair-like structure, and that's why we call it trichus. So the bacteria, if the bacteria do not have flagella, then such bacteria is known as Atrichus. Okay, no flagella. Example, all coccus, all the spherical bacteria, all the coccus or cocci, they never have flagellum or flagella. Flagellum is singular, flagella is plural. Okay, so that all the cocci are Atrichus. Getting it? Great. What if there is one flagellum present on a bacteria? Then we call it monotrichus. Then we call it monotrichus, right? Mono means one, trichus for flagella. So if there is one flagella on bacteria, it is monotrichus. Let's see the example. Pseudomonas, okay, Thiobacillus. Now, both of these organisms are important. Do remember Pseudomonas and Thiobacillus we will study when we will study the nitrogen cycle. Okay, these two bacteria are helpful in nitrogen cycle. Okay, great. That is something for later. What if a bacteria has single flagellum on either side? Okay, we call it amphitrichus. You know, the word amphi means both and trichus means hairs, right, or flagella here. So if the flagella is present on both the sides, single flagellum, then such bacteria or such condition is known as amphitrichus, right? One flagellum on either side. What is the example? Nitrosomonas. Okay, Nitrosomonas. Great. Now, what if instead of one flagellum at one end, there is a bunch of flagellum? Okay, a bunch of flagellum on one side of bacteria. Then such condition is called cephalotrichus. Now, the bunch of flagellum is also known as tuft of flagella. Okay, tuft is bunch. So if there is a tuft of flagella on one side, such bacteria is known as cephalotrichus. Okay, now example is Corynebacterium. Okay, next type of bacteria is Lophotrichus. Now, what if a bunch or a tuft of flagella is present on the either side of the bacteria or on the both side of bacteria, then such condition is called Lophotrichus. What is the example? Salmonella. Okay. Now, apart from that, the last type is Peritrichus. Okay. Now, peri means all around, or peri word comes from perimeter. Okay. So, Peritrichus, in this condition, flagella is present all through the body or all on sides of the body. Example, E. coli. Okay. So, E. coli has flagellum on all sides of its body. Okay. So, on the basis of flagellum, the bacteria can be Atrichus, Monotrichus, Amphitrichus, Cephalotrichus, Lophotrichus, and Peritrichus. Okay. Great. So, what we saw is bacteria on the basis of their shape, that can be coccus, can be bacillus, vibrio, or spirillum, and on the basis of flagella they can be Atrichus, Monotrichus, Amphitrichus, Cephalotrichus, Lophotrichus, and Peritrichus. Okay. So this is it for this video lecture. In next video lecture we will continue and we'll learn more about the bacteria. Okay. So stay tuned and stay safe. Thank you.